Mycoplasma Detection by Riano and Gareau. Mycoplasma is a common contaminant found in tissue culture laboratories, so proper aseptic technique needs to be followed in order to avoid any contamination. Here, prior to our experiment, we can see that Eddie is cleaning the laminar airflow hood with 10% bleach and then 70% ethanol. That's right, get up high. Samples of cell media that are suspected of being contaminated are aliquoted into Eppendorf tubes. These tubes are gently centrifuged in a tabletop centrifuge to pellet any cellular debris. Be sure to load them properly and then close the top. The supernatant is now placed into new Eppendorf tubes, leaving behind that debris. This is done to all of your samples. Did you catch that? Be sure to discard your pipette tips in the solid biohazard bin. Just a few more samples. Always make sure your samples are properly labeled prior to centrifugation. The samples will now be spun at 20,000 xg for 10 minutes at room temperature. Wait a minute. That's right, put in that locking cap. This is to pelletize any mycoplasma that's suspected in the media. Now we're ready to run. After centrifugation, the supernatant can be carefully poured off into the liquid biohazard container, making sure to note and keep any small pellet in the sample tubes. Just a few more samples now. Good job, Eddie. Any pellet that is in the sample tubes will now be resuspended by adding 50 microliters of buffer solution. This is added to all of our samples. Now, we're going to heat our samples to 95 degrees Celsius for 3 minutes. This can be done in a simple hot water bath of a beaker with heated water and a foam sample holder. Ah, yes, that feels great. Yep, 95 degrees. We will now begin to prepare our PCR tubes by adding 35 microliters of water, to all the tubes, 10 microliters of reaction mix, and 5 microliters of our test samples. A positive control is also created at this time. The PCR reaction has primers that will anneal to the 18S rRNA of any mycoplasma present. The amplicon of the PCR reaction has a size of 270 base pairs. That's what we're going to be looking for in gel electrophoresis. Just a few more now. There's the positive control. Okay. Now it's time to make our 2% agarose gel. This is done by carefully weighing out 1 gram of agarose into a weigh boat. Place that in our Erlenmeyer flask. And add 50 milliliters of our running buffer and give it a good swirl. The solution is microwaved in order to melt the agarose into the running buffer. Our mixture now melted, it's time to add ethidium bromide to our liquid agarose. This is so any bands in our gel can be visualized in the UV gel dock. Once the agarose has cooled slightly, it can be poured into a gel casting tray and left to solidify. While that's happening, we can begin our PCR reaction. Always be careful to label your samples. And remember their orientation as you place them inside the thermal cycler. Just one more. There we go. The Akron protocol for the PCR is to be followed, and we're lucky to have the Mycoplasma test already saved. Wow! Now it's time to run our PCR products by gel electrophoresis. A base pair ladder is loaded to our gel to make looking for the 270 base pair product easier. 20 microliters of each PCR product, those are our test samples, are loaded into the gel, including the positive control. Run the gel at 100 volts for approximately 45 to 55 minutes. And here's our results. All that can be seen is our DNA ladder and primer dimers at the bottom of the gel. Our positive control is even missing its band at 270 base pairs. What happened? Thank you for watching, and thank you, Dr. G. Made by Kevin Gareau and Eddie Riano.